Okay. So before we start with the presentations, I think Deborah is going to introduce the SHIN grant program for the Native Plant Society.
Yes.
So Deborah is our current chair of the, is it the Grants Committee? Scholarship
Grants
Committee.
and Scholarships Committee. Yeah.
For the North Carolina Native Plant Society. So this is a neat little grant that they have for students who are doing projects on native plants. So Deborah, if you want to introduce that program.
Yeah.
The deadline is coming up really soon.
Yes. So thanks to Lynn and Katrin for organizing this excellent seminar hour and giving us a chance to hear from our 4 speakers, hear about their interesting work. And all 4 of these students receive awards from the Tom and Bruce SHIN Fund, administered by the North Carolina Native Plant Society. We are pleased to support student research on North Carolina's native flora. And every year we award several grants to undergraduate and graduate students. So the SHIN Fund was named in honor of Tom and Bruce SHIN, their husband and wife team. They were native plant enthusiasts who did extensive gardening with native plants back before people were really interested in such thing. And because of this, they had to develop many plant propagation techniques. This was in the 60s and 70s, and these techniques were eventually turned into a manual, and then the proceedings from this manual became sort of the seed for the SHIN Fund. And nowadays, we maintain the fund through plant sales and fundraisers. And I do want to thank any donors in the audience for continuing to support this valuable resource for students. So the SHIN Fund is open to any university or college student researching basically any aspects of native plants. So if you're a student interested in applying to the fund, I provide a link in the chat to our website, and that's where you'll find all the relevant information. And others in the audience might be just interested in browsing about past recipients to see what they've been working on, and you can get to that information as well through that same link. And importantly, advisors, please pass on this information to students who are eligible to apply. And the deadline is coming up very soon. It's March 1st. So I'm hoping that there will be lots of people interested in applying. And I look forward to hearing from today's speakers. And I'll turn it back over to Ann-Katrin.
Okay, thanks, Deborah. So we have 4 student speakers today from a variety of universities. The first one on my list is Vanessa Gremler, who actually happens to be my student. And so Vanessa came to the lab interested actually in medicinal fungi. So she's working on a project looking at endophytes in one of our native plants, the shining club moss, Huperzia lucidula. So I'll turn it over to Vanessa to tell us about her research. I think you have to unmute yourself.
There we go. Hi, everyone. My name is Vanessa Grumler. I am an undergraduate student in Dr. Rose's lab, and I'm going to tell you about my project entitled Analysis of AChE Inhibitors in Huperzia lucidula Endophytes. Now first, we will cover some background information regarding the plant of interest as well as the inhibitor we were testing for. So this is Huperzia serrata, or the Southeast Asian toothed club moss. It is in the Lycopodiophyta division and the Huperziaceae family. It is a slow-growing club moss that reaches a height of somewhere between 3 to 11 inches at maturity. Huperzia serrata is native to Southeast Asia, specifically Japan, China, Tibet, and the Korean Peninsula. Huperzia serrata has been of particular interest over the past 30 years because it has been identified as a producer of an alkaloid called Huperzine A, or Hup-A. Hup-A has been found to act as an inhibitor to the acetylcholinesterase, or AChE enzyme, and the inhibition of this enzyme has been found to be effective in the early treatment of Alzheimer's disease. The treatment of Alzheimer's disease with HupA was brought to the public and supplements began being sold on the market in 1995. The use of Huperzia serrata for Alzheimer's treatment has resulted in a decline in the AChE serrata populations. This is due to both unsustainable harvesting practices and the slow growing rate of AChE serrata. This decline has prompted the need for research on supplementary sources of Huperzine A. Now, in the search for other sources of HupA, a study from 2005 attempted to analyze other sources of HupA in the Huperzia family. In this analysis, they found several other species of club moss that also produce HupA, including our plant of interest, Huperzia lucidula, which is native to North Carolina. Here we have our plant of interest, Huperzia lucidula, or the shining club moss. H. lucidula was found to also produce the AChE inhibitor HupA, making it a possible alternative source for this alkaloid. However, it is also a slow-growing club moss that only reaches about 6 to 8 inches at maturity. Due to its slow-growing nature and small size, we assume that if it were heavily poached, it may suffer the same fate as the exhausted populations of H. serrata. You can see that Huperzia lucidula has a wide range across North America, including areas in Georgia all the way up to Canada. Several years after the Huperzia family experiment, in 2013, another study was searching for alternative HupA sources and found that endophytic fungi living within Huperzia serrata was also capable of producing HupA. Now, endophytes are bacteria or fungi that live symbiotically near or within healthy plant tissue. Unlike pathogens, the presence of endophytes is harmless or even beneficial to the plant. On the right here, you can see an example of endophytic mycelia living within an orchid species. Some endophytes are capable of producing identical or similar secondary metabolites to their host plant. An early example of this research can be seen in a 1993 study where an endophytic fungus was found to synthesize the same cancer treatment ingredient Taxol as the plant it was isolated from. Now, although Huperzine A has been found in the North American shining club moss, H. lucidula, no research has been published identifying which of its endophytes, if any, produce the sought-out enzyme inhibitor. So our objective is to isolate endophytic fungi found in Huperzia lucidula and identify their acetylcholinesterase enzyme inhibiting abilities. Now that we have discussed the background behind our interest in the endophytic fungi within Huperzia lucidula, I will describe the methods behind our experiment. We began by collecting live plant samples from the Appalachian State University Nature Preserve and the Tater Hill Plant Preserve, with permits and guidance from Dr. Estep, who is a steward of the Tater Hill Plant Preserve. The isolation of the endophytes began by using a stepwise surface sterilizing of the plant material. The surface sterilized plant material was then placed on potato dextrose agar plates to promote fungal growth. Then the endophytes were given time to grow, and the initial endophyte plates had many endophytes per plate. So we continued to isolate the endophytes by trying to take small samples from the margins of what looked to be a single morphotype. This process was repeated as necessary until we had a single morphotype per plate. Here are just some examples of the various isolated morphotypes. You can see they range in color, size, and shape. Once the morphotypes were isolated, we collected morphological information from the endophytes to aid in the process of identification later. We recorded average growth rate and area of zone of inhibition tests. Now, at this point in the experiment, we have isolated over 50 endophyte morphotypes. We have made ethyl acetate extractions of the different morphotypes and will use these to analyze their biological activity through an AChE enzyme inhibitor assay. The endophytes that pass this assay, indicating that they inhibit the AChE enzyme, will then be processed through high-performance liquid chromatography to confirm and quantify the presence of HupA. Following the quantification of HupA in the endophyte strains, we will identify the strains through DNA sequencing using common fungal primers ITS1 and ITS4. The results of this project can be used in future research to indicate target endophytes that can be considered for isolation and extraction of Huperzia A. Although the North Carolina native AChE lucidula is not regularly poached for Hup-A, proactively finding alternative sources of Hup-A may prevent this species from becoming a target if populations of AChE serrata become exhausted. We are grateful for the following grants: the Tom and Bruce Shin grant from the Native Plant Society, the University Research Council grant, and Office of Student Research grant, both from Appalachian State University. We would also like to extend a special thank you to the Tater Hill Plant Preserve for allowing us to collect plant samples, and Dr. Estep for facilitating that process. We also appreciate Drs. Culpepper, Shaw, and Venable for supporting this project by offering instrument use and assistance. And that's pretty much all I have for my presentation. If you all have any questions, I'd be happy to answer them.
Thanks. I think there's time for questions.
Hi, Vanessa, it's Dr. Tuberty. I might have missed it, but did you mention where you're able to find the populations of the club moss here in Boone?
Yes, we actually collected from the Appalachian State Plant Preserve just behind campus, as well as at Tater Hill with help from Matt Estep, or Dr. Estep, sorry.
So that's a pretty common distributed species then.
Yeah, we were able to find it pretty easily and in large groups.
Okay, excellent. Thanks.
Vanessa, Dr. Estep here. Just curious, did you find different isolates from the preserve versus what you found on Tater Hill, or do they all kind of look the same? Or do you even have the ability to answer that question at this point? I'm not sure.
We do have some that, from a morphotype perspective, they look similar, but you could also see some morphotypes that were pretty different between the two. So I was really happy to be able to collect from both locations.
Yeah, and you can maybe also talk about the DNA comparison that we're planning to do.
We were planning on hopefully doing community studies as well to compare the fungi that we collect from both locations to see if we see any differences between the two.
Hey, Vanessa, could you talk a little bit about the assay that you used, the bioassay?
We ended up purchasing a enzyme assay from a company that was already made, and then we are able to purchase the enzyme from an electric eel, which is, you know, not quite the same as the enzyme that we have in our heads, but it's supposed to be a pretty equivalent enzyme to be able to be used in the assay. I made ethyl acetate extractions of all of the fungi. And then we will be using a plate reader from Dr. Venable's lab to see the changes over about a 15-minute time span.
All right, thank you.
Any other questions? If not, I think, thank you, Vanessa. And if, we'll then move on to the next student on the list, who's Laura Hammond. And Laura, are you here? I think
Yep,
I've seen
I'm
you.
here.
Yeah, okay. So Laura is from North Carolina State University. Her advisor is Dr. Rebecca Irvin, and she's working on the question, who pollinates Venus flytraps? And that's actually an interesting question for me because I'm growing Venus flytraps at home and I've hand pollinated them. So for mine at home, I'm the pollinator. It would be nice to see here who actually is doing it out in nature. So Laura, please tell us about it.
Well, you've answered my question then. It's Ann Catron, so my research is over. But I'm Laura Heyman. I'm a fourth-year PhD student at NC State University. I'm co-advised by Dr. Irwin and Dr. Sorensen. And I'm going to talk about some real preliminary results from some work that I did in 2020. So the unique thing about carnivorous plants and their pollinators is that there's this thinking that perhaps these plants experience conflicting pressures between attracting two suites of insects as pollinators and insects as prey. And is there some potential conflict between that overlap? So here are two examples of carnivorous plant species where the authors did find some evidence that the plants are eating their pollinators to some degree, and that could potentially have a detrimental effect on overall reproduction. So my focal plant is a very fun Venus flytrap. That's my favorite flytrap quote there. Carolus Linnaeus told it to the plant's original describer, John Ellis. He said he denied the thinking that Venus flytrap could be carnivorous and said such a thing was against the order of nature as willed by God. I think that's just a fun, very metal quote. Numerous papers on the way that the trap closes and digests prey, but surprisingly, there are fewer on the ecology of the species in the wild. And that's just a fun GIF there, closing on a fly. They actually rarely eat flies, but I like that GIF because it's from a horror movie, so it's a bit of fun. So Venus flytrap has a very restricted native range. It's only natively found in North and South Carolina. It's been introduced elsewhere, but in North Carolina, it's found within about 100 miles of Wilmington. And in my range map here, those counties that are highlighted in various shades of green are places where Venus flytrap is extant. The deeper the green, the better the best population in that county. And those yellow, orange, and red counties are the places where it's been failed to find, or is historic, or has been extirpated respectively. So it's threatened in its native environment by fire suppression, habitat change, and illegal collection. So it's becoming rare. And it's pollinated by a generalist suite of insects, chiefly sweat bees and beetles. And this is from a study that was done in 2016 and 2017, was published in 2018 by a group of folks, including Dale Suter, who I think is on the line. So they determined that these were some of the most frequent visitors to flytrap flowers and were carrying flytrap pollen. And from some of my work, we do know that the seed set of Venus flytrap is in part limited by the effective frequency of these pollinators. So when you supplement pollen to a Venus flower to sort of drench it with as much pollen as possible, you end up seeing a slightly increased seed set, which might mean that the reproduction of this flower, of this plant, is limited by its pollination. Something also interesting about this plant, when we think about insects and how they relate to the conservation of this plant, is that sometimes management for insects and plants can differ. So for example, on the left is an extremely rare moth that specializes in eating Venus flytrap. And like its host plant, it obviously would benefit from regular fire, but unlike its host plant, its strategy is a little different. It doesn't hunker down and the fire from an underground route. It depends on a meta-population strategy where fire is hopefully patchy, a population from elsewhere will move in to where fire has been. So those management requirements are a little bit different between these two species. So my sort of main questions delve into the implications of pollinators for the conservation of this species, and also a little bit about why it's so good at not eating its own pollinators. So I've divided my methods here into two separate sections. First off, I'm interested in understanding who are the best pollinator taxa, who is the best at getting pollen from stigma, because we know who the frequent visitors are, but that doesn't necessarily always equate to who is doing the best job, so to speak. And before I get into a little bit here, I do have a brief
Let's hope she comes back.
Yeah, it seems we have the audio stuck right now. Technology.
Come on, I'm on the edge of my seat.
Well,
This seems to have been a technical issue.
she might try to come back in.
Yeah. Yeah, meanwhile, if anyone is interested in a Venus flytrap, I've been growing these things in tissue culture with students, and I have literally still hundreds of them. I'm going to sell them at the next plant sale at the Daniel Boone Native Gardens.
Do you also have tips on how to care for them? Because they're kind of
Yes.
hard to care for.
Yeah, they come with a care sheet. And I've been able to pollinate them and get seeds. So I was pretty excited about that. But I've hand pollinated mine. There was, I had a couple plants set aside to see what they're doing, but there were no seeds on those. Whichever pollinators we're talking about here, they're not flying around Boone.
Do you want to go on to Jessica and
We could and maybe see if then
Laura can pick up?
Laura
if
comes back
Yeah.
after. Oh, there she is.
Oh, there you are.
Hey, I fell out there for a little bit.
Yeah.
Yeah.
we were missing you.
My internet gave out. Should I keep going or should
Yeah.
Yeah, why don't you keep going? Yeah.
Okay. I will press forward. Sorry about that. Okay. Okay, so I think where I fell off, forgive me if you missed a little bit of it, but I bagged the flowers while they were in bud. I took the bags off, and then after a single insect visit, I would capture and squash the stigma in order to count the number of pollen grains deposited on the stigma. Quite tedious, but sitting in the field, so that's fine. Here's some preliminary results from 2018. We mostly saw visits from a lot of honeybees, more honeybees than I would want because they're not exactly the native pollinators. Other bees, lots of green sweat bees, and these bees seem to be doing a relatively good job, but we need more observations in order to sort of differentiate these different taxa. In 2020, my updated sort of continuation of these observations, I was excited. We got a lot of bumblebee visits. We also got a wasp and a buckeye. And what was really interesting to me is though we think that green sweat bees seem to be the most frequent visitor, I was noticing that bumblebees drag their bodies across the entire flower, while the sweat bees were sort of more differentiating between the anthers and the female parts. So I have a suspicion that bumblebees might be doing an excellent job here, more sort of outrepresented by how many visits they are. So I'm excited to analyze these data and have more concrete things to say about this. The next question I delved into from this summer was how do both resource and pollen limitation affect reproduction in the species? To do this, I have 3 different versions of this trial, but I'm going to speak to one specific version of this. I supplemented dead crickets to Venus flytraps starting last summer. I did this about every 2 to 4 weeks. I added a dead cricket to each open trap to those that got supplemental prey. And then a number of them in 2020 got supplemental pollen. And then I compared those different treatments to each other. And then a quarter of those unlucky plants got nothing. And so I supplemented these things and then I compared the seed set between flowers. So here's some preliminary results from that trial. So on the y-axis there's the mean number of seeds per fruit for these plants. X-axis is our treatments, prey, pollen, no prey, no pollen. And there, so far, it seems like there's actually no significant difference between all of these treatments, unfortunately. But I'm still working on the analysis a little bit and will point out that both those prey supplemental treatments have had a slightly higher, at least to my eyes, seed set than their associated non-prey treatments. So I'm excited to sort of get into the nitty-gritty of this data a little bit more. Some takeaways from this study from this past summer is that those sweat bees and beetles seem to be the most frequent visitors. it still remains unclear who is the best at getting pollen to the stigma and therefore might be of higher conservation priority for this species. Also, Venus flytrap reproduction may not be dramatically limited by pollen of prey limitation, at least within the timeline of this study, which has some possible ramifications for whether or not Venus flytrap truly experiences or suffers from this overlap in prey and pollen. It seems unlikely, but it's still fun. So future directions, more pollinator observations, love to get some beetles. I saw a beetle visit once right outside of my experimental flowers. That was a bummer. And some additional analyses. So I've done 3 versions of this trial, counted number of flowers, number of traps. I've been following folks, by folks I mean plants, for months and months. So I've got plenty of other stuff I'm excited to get into. And with that, I will thank all these folks, including obviously the North Carolina Native Plant Society, for helping to fund this work. And I appreciate it. Sorry for falling out there.
Thank you. That was interesting. And I think we have a couple of minutes for questions.
Hi, Laura. Could I ask a question about compatibility? Is the Venus flytrap self-compatible or is it incompatible?
It is self-compatible, but it still needs a mobile vector to get that pollen from one flower to another. It doesn't sort of spontaneously pollinate itself as a last resort.
Yeah, what I've noticed is,
oh, go ahead.
Sorry. What I've noticed with mine is that the stamen and stigma are mature at different time points, and that's probably how they prevent self-pollination. Is that, Laura, does that match your
That's true,
observation?
yeah, it's protandrous, those male parts develop first.
So one last question, Laura, I'm sorry. Do you see a difference when they're self-pollinated versus outcrossed in terms of the number of seeds produced or the size of those seeds?
That's a great question. And in fact, that's something I also did this summer, which I didn't present because I thought it was not exciting enough, but I did compare.
It's exciting.
It's exciting to me, but so individuals that I pollinated, so there's no significant difference in the seed set between those that were pollinated from another individual versus pollinated from another flower in the same individual. But there was a difference in seed set between flowers that were pollinated from within the same flower. So they did better if that pollen came from a different flower, whether it came from the same individual or a different individual. Basically, fresh pollen is best.
Thank you.
This is a really simple excuse me. This is a simple question. The moths eating the Venus flytraps kind of hit me like a Mack truck. Like, well, of course they get eaten too. I always thought of it as the other way. Do you see a lot of insect herbivory of the Venus flytraps?
You do, more than you expect. I don't think they're especially delicious from a nutritional standpoint. They're high in tannins. So sometimes they get gnawed by orthopterans, crickets, and grasshoppers, and by slugs, but I don't think it's anyone's favorite food except for this moth.
Cool, thank you.
I was wondering if you've ever seen a bumblebee getting caught. I would imagine they're too big, but
I saw a bumblebee half in, half out, not alive in a trap before, yeah.
That's pretty amazing, because those are strong bees.
Yeah, for sure. Maybe she was a weak bee. I don't know. Had a bad day. Cool. Thank you.
Okay. Thank you. So good job. If there's no more questions on this, then we'll move on to the next project. So that's Jessica Roach. Jessica, are you here? Yeah, I see.
All
see
right.
your name. So Jessica is from the University of North Carolina in Wilmington. Her advisor is Darren Pennies, and she's working on the endangered rough-leaved yellow loosestrife and doing genetics analysis on that one. So Jessica, I'll leave it up to you to tell us about your project.
Thank you very much. Can everybody hear me okay?
Yes.
Okay, great. I was having issues with my camera earlier, but now that seems to have subsided. So Thank you for allowing me to speak today at this seminar. This is very exciting. I'm very happy to share what I'm working on, and I'm very excited to have received the SHIN grant. My thesis work will look at the extent of genetically unique individuals between and among extant populations of Lysimachia sperlifolia. This is to confront issues regarding life strategy and reproduction. My advisor is, as we have already gone over, Dr. Darren Pennies, and my committee members are Drs. Stephanie Kamel and Johnny Randall. There we go. All right, fun stuff. So as we know, the Southeastern Atlantic Coastal Plains region is recognized as a globally biodiverse hotspot. This classification is defined by having more than 1,500 described endemic plant species confined to a region with greater than 70% habitat loss. The coastal plain floristic province lies within the geological southeastern coastal plains and is host to several ecosystems unique to this region, including the longleaf pine savanna. Longleaf pine savannas once dominated the southeastern coastal plains, covering roughly 90 million acres from Virginia to East Texas and through the southeastern United States. However, due to high anthropogenic impacts and disturbances, including lumber harvest, fire suppression, hydrologic change, agriculture, and urbanization, less than 5% of this historic range remains intact today, amounting to approximately 3 million acres. Higher than expected rates of plant endemism and rarity occur throughout this region, and as the name longleaf pine savanna suggests, stands are dominated by Pinus palustris, while the herbaceous layer is characterized by the presence of Aristida stricta and associated graminoids. There is abundant diversity in this herbaceous layer, and there is abundant diversity in the herbaceous layer of these savannas, where 1 square meter can host up to 50 different plant species. Within this highly diverse herb layer exist some of the most charismatic and recognizable plants, including 5 genera of carnivores. And even though this is not what I'm doing my research on, perhaps the most notable is Dionaea muscipula, or the Venus flytrap, which is often found along ecotones of longleaf pine savannas and picosins, which are just upland wetlands. Flytraps are narrowly endemic species only found in the coastal plain of North and South Carolina. They thrive under strict habitat requirements, including mesic, nutrient-poor, acidic soils with frequent fires and an open understory. The current consensus indicates an approximate range of about 75 to 100-mile radius around Wilmington, encompassing 18 North Carolina counties and 4 South Carolina counties. These insectivorous plants are listed as imperiled at the state and global level, and is of special concern vulnerable in North Carolina. Though no federal protections currently exist for this species, a formal application submitted in 2016 is currently under consideration. But what about the other plant species, right? We have tons of variety here. While Dionaea muscipula is a fascinating plant with animal-like behavior, our amazement with its unusual habits may overshadow the importance of many other and perhaps less charismatic, but even more imperiled plants. The species of focus for my research is Lysimachia sperlifolia. This is commonly called the rough-leaved yellow loosestrife, and this loosestrife is also a narrow endemic, commonly found along the ecotones of longleaf pine savannas and picosins. It shares nearly identical habitat requirements to the Venus flytrap, and observation of one species is often indicative the other is nearby. Lysimachia sperlifolia has known populations in 11 counties across North Carolina and 1 county in South Carolina. This loosestrife is listed as endangered under the Endangered Species Act, vulnerable in North Carolina and globally, and is imperiled in South Carolina. Federal protections and active monitoring programs have been in place for this species since 1987. It's longer than I've been alive. So not only do these plants cohabitate, but they also share similar life habits, right? So both flytraps and loosestrife are clonal organisms. They spread through underground stems called rhizomes. Rhizomes in these plants are a presumed adaptation to the frequent fires that occur in this region, which in combination with low nutrient availability, maintain a low shrub layer and prevent a hardwood understory from developing. These rhizomatous species produce vegetation rapidly after burning, and fire is known to promote flowering in flytraps and other fire-adapted plant species. This is not the case for loosestrife. In fact, loosestrife does not typically produce flowers, and when it does, fertilization events are rare due to a lack of effective pollinators and suspected sterility and/or S allele incompatibility. So S allele incompatibility occurs when the genotype of pollen matches with one of the S alleles of the pistil. Self-incompatibility is actually an effective mechanism that promotes outcrossing in plants and is the botanical equivalent of avoiding incest. Thus, those self-incompatible specimens with the same S alleles are incompatible, say that 10 times fast, and are allocated in the same incompatibility group. So put a little more simply, loosestrife is a clonally dependent organism with an inexplicably poor sexual reproductive strategy and has not exhibited population increase nor stability in monitored populations over the last 30 years. Results from the 10-year U.S. Fish and Wildlife Service monitoring and management plans has shown an overall decrease or undetermined status of these populations with a population viability analysis using Bayesian state space models. And previous studies have investigated reproduction, fruit and seed dispersal, and seedling establishment based on Mendelian genetics, though no clear explanation for the lack of successful sexual reproduction has been found. Lysimachia spirifolia has been federally protected for over 3 decades, yet little understanding of the species itself and how best to manage it has been gained. A population genetics study would be a first for the species and critical for future management practices where genetic bottleneck is of serious concern. So, this study will use 11 unique microsatellites to determine the genetic diversity among extant populations. And microsatellites, as we may already know, are highly variable, non-coding repetitive DNA sequences, 1 to 6 base pairs in length. The variations in these repetitive sequences allow information about an individual to be inferred from other members of the same population. So, leaf tissue samples will be collected from 7 of the 9 known populations in North Carolina and 1 in South Carolina. Sequence DNA from a separate study of the population genetics in specifically the Fort Bragg loosestrife population will be shared by that team's PI. And in the field, a preliminary stem count to confirm a minimum of 20 stems with leaves and whorls of 3 or greater will be conducted. This is based on the understanding that stems with 2 opposite leaves are presumed immature and therefore cannot be sampled. I'll be grabbing a single leaf from each stem and desiccating in silica gel and storing in a dark, cool container. The molecular work will be conducted in the Penny's lab, where we will extract genomic DNA using QIAGEN DNeasy Plant Pro Kit and amplify using QIAGEN Multiplex PCR Kit with custom fluorescent primers. And subsequent data will be sequenced and analyzed using Mega, Structure, and R software. This population genetic study of Lysimachia spirifolia throughout its natural range is expected to produce data that will be instrumental in producing the most comprehensive management strategy to date. Future studies will be able to apply results from this research in situ and to ex situ conservation efforts and obtaining and preserving land with suitable habitat for reintroduction. Better understanding and protecting this plant will contribute to the maintenance of imperiled coastal plain ecosystems. And it is our goal with this research to do our part to ensure the exceptionally diverse character of wild North Carolina does not become a matter of historical record, because it's only fair that future generations are afforded the same opportunity to experience the dynamic beauty found in our very own backyard. That's all I have on my work, and if there are any questions, I would be happy to answer them.
Thank you for the presentation. That's an interesting problem to have for a plant, right?
Very much so.
Hi, Jessica. Could I ask you a question about those the self-incompatibility? Do you know if that's a gametophytic or sporophytic system?
Do I know if it's a gametophytic or a sporophytic system?
They're the 2 types of incompatibility systems with S loci. It's okay.
We don't know yet. Nope,
I gotcha.
we
Is
don't.
the species a diploid or is it a higher level polyploid?
It is diploid,
Oh, so that'll make it nice and easy.
hopefully.
How many samples do you expect to collect across the range?
It would be ideal to get around 200 samples. And right now I've got 60 between two different locations, Boiling Springs Lake and Croatan National Forest. It's been a little difficult getting onto the majority or into the majority of populations because most of them are actually on military bases. So there's been a lot of extra red tape since the pandemic has gone full swing. But 200 ideally, and at the very least, at least 100 samples. But I'm encouraged that once some restrictions lighten up a little bit, we'll be able to hit our goal.
Thank you.
Thank you.
So I was wondering, once you identify different genotypes in there, is there a plan to then maybe transplant some plants or cross-pollinate to give them some more genetic variation?
Right. So we're kind of hoping that this could be the stepping stone for that, right? To encourage future research. As many of you may know, Misty Buchanan at the Natural Heritage Program, she is essentially the global expert on this species. She did her thesis research on it. So there actually hasn't really been active research since about 2001, 2002, and we're trying to get some momentum again. So this would be a pretty good baseline, we think, for future studies.
Are there more questions for Jessica?
Thank you all so much. I appreciate
Thank you. So then our last speaker here is Ben Brewer. I see, Ben, you're here. You're done with your genetics lab. So, and Ben is actually working on a plant that I've been going out and photographing for quite a few years now. And that is Grace Lily. If you've ever been up to the Rhone Highlands around the July 4th weekend, you know that plant. It's a really beautiful red flower, yeah, right there. And it has some issues with fungus and I think some hybridization going on. So Ben is going to talk more about that, hopefully. So Ben, I'll leave it to you to tell us about this project.
All right. Yeah. First of all, I just want to say what a great lead in to me. Everybody has done awesome. And hello, I'm Ben Brewer, but I am here to introduce the real star of this show. And that is Gray's lily, or Lilium grayi, which is one of our most stunning Southern Appalachian endemic species, and then talk a little bit about some of the work that I'm doing to try and protect it in as many ways that I can. So around here at App State, Southern Appalachian endemic is a term that we hear a lot because we do live in one of the biodiversity hotspots of our country. Our region is home to over 10,000 species, many of which are endemic, which is primarily due to a complex history of glaciation over the past 40,000 or so years. During the last glacial maximum, as glaciers extended southwards and covering into around Pennsylvania, northern species were forced to migrate southwards. Then, when the climate rewarmed and those glaciers retreated back towards the poles, some of those populations of species retreated north to follow them and restore their original ranges, but some retreated up into the mountains, seeking out those cooler climates at higher elevations. This, combined with the fact that our region was never fully glaciated, has created a region that is rich with species, but also inherently at risk to future changes in climate or land use. And that's what this map is showing. Produced by Clinton Jenkins and their team in 2015, they created a priority score for the country. And this priority score is a function of the number of endemic species found in an area and how preserved or protected their remaining habitat is. Areas with the red or orange are those with a high priority score. And as we can see, our Southern Appalachians were determined to be the #1 priority for future land conservation because of our threatened endemic species. Which brings us back to Gray's lily. which I think is emblematic as a Southern Appalachian endemic species. It was originally discovered on Roan Mountain on the North Carolina-Tennessee border, described as a species by Serena Watson in 1879. The plant is a fairly long-lived perennial species, taking between 3 to 7 years to become reproductively active. It overwinters as a scaly bulb, emerges in the spring, produces these gorgeous flowers in the early summer, and then the dehiscent fruit or dehiscent capsule, which is its fruit, in the late summer. And then normally in the fall, the plant will senesce or its above ground tissues die back, and it again overwinters as that scaly bulb underground. The species loves wet areas like high elevation bogs, seepages, moist forests, but it also really thrives in the more xeric, grassy bald plant community. Nearly all populations are found above 850 meters in elevation, but this goes all the way up to nearly 1900 meters atop grassy ridge bald at Roan Mountain. NatureServe lists the species as critically imperiled. It is a federal species of concern, and it is listed as threatened, endangered, and imperiled in North Carolina, Tennessee, and Virginia, respectively, which are the only 3 states where it occurs. The primary threat facing the species is the early season population collapse or premature senescence due to the invasive fungal phytopathogen Pseudocercospirella inconspicua, which causes lily leaf spot disease. Now, remember, originally in the fall is when they senesce and die back after producing their capsules. This disease causes them to wilt and prematurely senesce, oftentimes in the summer before they're able to successfully reproduce. This phenomenon was originally observed by Moni Bates of the North Carolina Plant Conservation Program in the late 90s. She did some great work monitoring populations at Bluff Mountain in Ashe County and Sparta Bog in Allegheny County. And then the Leedy Lab out of Eastern Tennessee State University has done some really great work with monitoring at the large population up on Roan Mountain. And this includes the master's thesis by Russell J. Ingram, who was able to confirm the identity of that pathogen as Pseudocercospirella inconspicua. Additionally, in that lab, they did a pretty extensive search of herbarium records to try and figure out when we think this invasive pathogen first showed up here in North America. They were able to find records of it in a Lilium canadense individual from Vermont from 1916, and their earliest record in Gray's lily was from Roan Mountain in 1947. Kind of carrying on the torch, Cindy Barrett, also of that lab, recently finished her master's thesis where she went through to multiple populations across the range and confirmed that it was Pseudocercospirella inconspicua affecting those populations as well. So here we can see physically what the disease looks like. It's these brown or yellow lesions on the leaves, and eventually these will radiate throughout the plant and cause the entire thing to wilt. We can see some leaves a little further on in the process down here at the bottom. Again, thanks to Russell Ingram, we know that this is caused by fungal canidia, which we can see here magnified at 90 times magnification, fungal canidia on the Gray's lily leaf. So this prior work has really been critical and instrumental in determining the problem. And what we really need now is to figure out how populations across the entire range of the species are doing, which brings me to the goal of my research work. I am conducting a range-wide health assessment of as many Gray's lily populations as possible in 2 primary ways. First, by conducting a genetic diversity analysis, where I will use microsatellite markers to get population health metrics such as heterozygosity and allelic richness. And additionally, we'll be able to evaluate population structure or interconnectedness of populations across the range. Simultaneously, I'm conducting demographic monitoring of these populations to try and estimate how many flowering individuals are actually successfully producing seeds and capsules. So to that end, last summer, I visited a total of 27 Gray's lily populations. I was successful in finding flowering individuals at 18 of them. On this map, the individual points represent individual plants, and the different colors represent different populations. So of those 18 populations, however, 6 of them, represented here as the diamonds, are those in Virginia that are thought to be experiencing introgression or hybridization from the sister species Lilium canadense. And this produces the hybrid Lilium pseudograyi. The other 12 Gray's lily populations represented with circles are those that we think are true, pure Gray's lily populations. Additionally, to try and investigate this hybridization, we also sampled from a population of Canada lily, represented here with the red square, located further south and at a lower elevation than all of the known graze lily populations. In total, I sampled 350 individuals for this genetic diversity analysis. My monitoring protocol was developed by Leslie Stark of the Plant Conservation Program, Dr. Chris Ulray of the National Park Service, and Dr. Matt Estep from here at App State. And the idea here is to monitor populations over the course of 2 visits. First, in the early summer when plants are producing their flowers, and then a couple months later in the late summer when their capsules are maturing and their seeds are about to hiss. So on my first visit, I collected some general plant health metrics, including the height of the plant in centimeters, the number of leaf whorls on the plant, defined as 3 or more leaves, so we would not count this little single leaf right here. How many of these leaf whorls show signs of lily leaf spot, which is again this yellow and brown lesioning on the leaves, and then some data about the surrounding vegetative cover, both from the ground up to 1 meter and from 1 meter up to 2 meters. On these first visits, I fixed each tag with an aluminum tag for, fixed each plant with an aluminum tag for identification. I took a GPS point and I sampled a small bit of leaf tissue for the genetic diversity analysis. Then I returned a couple months later to rediscover individuals and determine whether they successfully reproduced, senesced the disease, or if something else happened. So these are the results from my first visits to these populations. All of these results are averaged across each population. In total, I was able to tag and measure 225 plants. Across all populations, average height was slightly less than 90 centimeters, 89.7. On average, just around 6 whorls per plant, of which slightly less than 24% showed signs of lily leaf spot. And then the average flowers per plant was 1.8 or slightly less than 2. I'm currently in the process of doing statistical analysis to see if there are any significant differences between these populations. These are the results from my second visit to the populations. In total, I was able to rediscover 207 of my 225 plants, or 92% of them, of which only 67, or less than a third, successfully produced capsules. A plurality of individuals, 74, were found fully senesced to lily leaf spot disease. A total of 48 individuals were found browsed or victim of herbivory. This includes 23 plants which were found browsed and senesced to lily leaf spot disease, and 25 plants that were found browsed but otherwise healthy. The final 18 plants did not produce capsules for miscellaneous reasons. This includes 11 plants that were mowed, so I'm not sure if they produce capsules or not, and 7 plants which were found healthy but with no capsule. I suspect lack of pollination. All of these values were calculated only based on rediscovered plants at each population, though I do suspect that most of the ones I was not able to rediscover did senesce to the lily leaf spot disease. Along with some other organizations, we did similar monitoring at 2019 at Grandfather Mountain and at Tater Hill with fairly comparable results to 2020. And this isn't great, but unfortunately, it does get worse when we remember that 6 of my populations are those that we think are experiencing that introgression or hybridization. So when we split out these 6 populations in the orange from those that we think are true Lilium grayi in the green, we see our flower capsule production rate plummet from 32% down to 22%. This is the result of both increased senescence to the lily leaf spot disease and increased herbivory, as we see senesced plants jump from 35% to 40%, senesced and browsed from 11% to 15%, and then purely browsed from 12% to 17%. Additionally, the fact that the hybrid populations are outperforming the pure Lilium grayi populations suggests there's a real risk of genetic swamping, where the genotypes and alleles that make graze lily what it is are at risk of being eroded by the hybrids and the Canada lily. So now that I've depressed you a little bit on the macro scale, I want to zoom in and talk about 3 of my favorite plants. This on the left is individual number 10441, on the right is 10440, and in the back is 10439. I love these plants for a couple of reasons. First, I think it's a little hilarious that I hiked over 150 miles in some of our most beautiful, high country areas, and then found the most spectacular plants on the side of the road a mile down from Carver's Gap. 10441 had 12 flowers, 10440 had 10 flowers. The next closest was 7 flowers on an individual at Little Hunt Mountain. So really, these are spectacular plants. But the real reason that I love them so much is that together, the three of them produced 26 flowers, all of which successfully matured into capsules for 26 capsules. This is more capsules than I found at any other entire population. Across all 18 of my sites, I discovered 128 capsules. So these 26 make up 20% of that entire total, all of which are right next to the road. As you may be able to see here on the right from my October photo, these plants were about 2 or 3 feet away from getting mowed. In fact, there was a fourth plant, one of 438, a little bit further up the road, that I did find mowed when I first revisited on the 11th of September. This is extremely concerning given their reproductive output. And I do think that these plants are likely at risk of being mowed every single year. Luckily, last week, I gave a similar talk at the Rhone stewardship meeting, and we're currently in the process of maybe having some conversations about the way we do mowing and maybe trying to reduce mowing on this approach up to Carver's Gap, or at least do it later in the year. So that was October. Now we move forward to today. I'm currently in the process of extracting DNA from my 350 samples using a modified CTAB method in preparation for microsatellite amplification via PCR. Ideally, I will be using 16 microsatellite markers to amplify the DNA in these individuals, but this will kind of depend on the success of those markers in amplification. Down here, you can see one of my more successful extractions so far, where I was able to obtain 140 nanograms of DNA per microliter of solution, which is about what we hope for. As far as demographic monitoring, I do plan to continue in the 2021 season at all of my 18 sites from 2020, and depending on permitting, I hope to be adding 3 more to that list. And I want to end just by talking about the extreme effectiveness of these signs placed up at Roan Mountain along the Appalachian Trail by the Southern Appalachian Highlands Conservancy, U.S. Forest Service, and Appalachian Trail Commission. We can spread lily leaf spot. We as humans can spread lily leaf spot with our touch. And so it is incredibly important to not touch or pick the flowers. And so when I was out there doing this work last summer, people ask you what you're doing. And every time I mentioned Gray's lily, they all said, oh, please don't touch or pick the flowers. They had read the signs, they knew what was going on and how they could help, or more importantly, how they could not hurt. And I think this is critical because everyone who's out there in our recreational areas, they're there because they love nature. They just don't always understand the damage they can do. So I think every ounce of education that we can get out there will have a massive amount of returns. And so I will just kind of finish by saying these signs are incredible, and I think they need to be placed up at every single Gray's lily population that is accessible to the public. And so I am hitting my time right now. So unfortunately, I don't have time to thank all the incredible people who helped me both in the permitting process and the fieldwork process. But I do want to make sure to thank Leslie Stark of the Plant Conservation Program, Dr. Chris Ulray of the Park Service, and of course, Dr. Matt Estep of App State for developing my monitoring protocol. Give a huge thank you to the Native Plant Society of North Carolina for funding through the Tom and Bruce Shin Grant and making all of this possible. And then thank the Office of Student Research for funding as well. And with that, I would love to take some questions and talk about lilies.
I have a very good presentation. I have actually 2 questions. One of them is more silly and one of them is serious. So you said it was identified and described by Susan Watson, if I remember her name correctly. So
Serena Watson. Yeah.
Serena. Okay, sorry, Serena. Why is it called Gray's lily if it was discovered by her?
Well, that's a great story. The hybridization issue has plagued the species since its discovery. When Asa Gray was the one who originally discovered the plant in 1840 on Roan Mountain, and he identified it as a Canada lily. He said it was Lilium canadense. And it wasn't until 39 years later when his student, Serena Watson, was looking at the plant, he was the one who really, determined that it was its own species. And so he named it in honor of Asa Gray, the esteemed botanist who made the wrong call the first time.
Okay, and my second question, the hybrids. Do the hybrids have any visual cues like that, like not just genetic, because I know you've done genetic work on them. Do they have any visual cues that might tell you, like on the field, that they might not be gray lilies and they might be a hybrid instead?
Yes. So hopefully we can see on this picture, okay. Basically, the main difference between the two, Gray's lily has nodding flowers that are at somewhat of a horizontal slant, whereas Canada lily typically are fully facing downwards. This is kind of one of those hybrid traits that as you go along the hybridization road or hybridization climb, you'll slowly see the Gray's lily characters diminish and the Canada lily characters increase. So one of them is the nodding amount of the flowers, and then the other one is how reflexed the tepals are. Gray's lily is really distinguished by having that campanulate or bell-shaped flowers where the tepals are just recurved right at the very end of the plant. Canada lily, basically it's all the way back. Or the tepals bend all the way back. So the anthers and stigma are actually exserted. If you look at the flower from profile, you'll be able to see the anthers and stigma sticking out of the plant. Whereas in Gray's lily, they would kind of be, in profile, they would be blocked by the tepals not being that reflexed.
Thank you so much.
Ben, I have a question about your favorite plants. Have you ever,
Yes.
are they part of a larger population along the road? And have you ever, do you find that fungal pathogen in this population or on these favorite plants?
No. No. There was a small amount of the lily leaf spot on them, but not the way we see it on other plants. My kind of pure speculation about this is that it all comes down to deer and herbivory. There is some work that's shown that herbivory and browsing can help spread these pathogenic diseases. Not necessarily in lily leaf spot, we don't know that for sure, but in other pathogenic diseases this happens. And so basically right off the other side of the road here, on the other side is a pretty steep drop. This is a fairly narrow road going up to Carver's Gap. And I think it's just deer have a much harder time accessing it. And so, yeah, in relation to other populations, I do consider them to be part of the round-balled population, but it is, I think, about 3/10 of a mile northeast from the other main cluster of round-balled. So they are separated off by a little bit.
Do you have any idea why the ones along the roadside there had such better seed production than all of the others in your study?
I suspect that they're very old plants and so have just been able to photosynthesize a lot and amass a lot of resources. And then my other pure speculation is just the deer, that because of their location, deer have a much, or at least herbivory has much harder time getting down there. And so the disease is less spread and they're less likely to be eaten. But again, that's more on the speculation side.
Hey, Ben, I wanted to ask you about capsules. By the way, good job on the presentation. When you said and those capsule percentages are pretty discouraging, would you say that those plants that were able to fruit had viable seed in them? Or because I know sometimes the capsule, it can fruit, but then the capsule itself kind of withers before it's able to produce seed.
For the most part, yes. I generally attempted to, in any capsules that I hit very early on that were still very green, I tried to revisit them and see if they did mature successfully. I don't have exact numbers on that, but the general trend is yes. I certainly saw some capsules that were clearly infected by lily leaf spot disease. And I do think, yeah, I basically didn't count those as capsules successfully produced because, you know, they'd be withered, very clearly not viable.
That's good. I'm glad to hear that. That would make me even more depressed if you counted those withered capsules. And do you think that, did you see any patterns in like habitat wise in terms of which plants were able to produce fruits? Like there's been a speculation that the grassy bulb plants can perhaps successfully reproduce because they're in a more open habitat than some of these shady kind of forested sites.
Yeah, I had some head scratching results there because that's that was my prediction going into the season. And certainly the grassy ball production or grassy ball populations were among the best, specifically round balled at 50% and little hump and hump mountain at 45%. But the thing that makes me really scratch my head is the next two most successful populations are those at Pinola Bog and Sugar Mountain Bog, which are very wet populations. And they had 41 and 42% successful capsule reproduction rate. So this is an area that I need to analyze my data a little better to give you a good answer. But I definitely did see very successful populations in wet areas, and that was strange to me. Granted, they weren't very shaded. Yeah, I mean, they were decently shaded. Yeah, I'm honestly not sure. Certainly, we saw excellent production on the balds. In general, poor production on the not balds, but then Pinola Bog and Sugar Mountain Bog are head scratchers. And then kind of similarly, Big Spring Bog up in Virginia. Again, a hybrid population, so we have to take it with a grain of salt, but this was another very successful population that was very shaded and very wet. So long answer to say, I'm not fully sure.
Well, maybe the cover biostrata that you collected will be insightful too. But thanks.
Yes. Definitely. Thank you. I didn't know you were here. I'm very glad you got to see this.
Me too.
Hey, Ben, since I asked everybody else, is Grace Lily self-incompatible?
Yeah, you better believe I was Googling because I don't know the answer to that. So there are other lilium species that are gametophytic self-incompatible. I do not know for Gray's lily, and I'm sorry that I'm a bad scientist.
Not a bad.
Yeah, I'm not sure. Sorry.
You did a lot of great work here. It's just there's lots of open questions that we need to be thinking about. I'm really pleased with your work, Ben.
Thank you.
Yeah, I find it funny that the best plants were along the road. I found the most beautiful plant I've ever photographed actually right behind the bathroom up on Roan Mountain at the parking lot. So it seems like in these areas, maybe where there's more humans, more cars, less deer, maybe that's where they do the best. So is deer browsing a huge problem there, or?
Yeah, actually, that was one of the things that really surprised me at Roan Mountain was kind of the difference between the three balds. You know, it's a relatively short area. I think it's 3 miles from Carver's Gap to Grassy Ridge Bald. But looking at the difference in herbivory, Grassy Ridge was a very poor population in terms of capsule production, but not really crazy in terms of senescence. The thing that really drove that lack of capsules was the fact that we saw, or that I saw, a lot higher herbivory up on Grassy Ridge. And again, speculation, I think that's just because of the remoteness. When I was up there sampling, I saw deer multiple times, and I think it's because Grassy Ridge is just the furthest from the trailhead. So yeah, I think that seclusion plays a big role for sure. Yeah, that was kind of one of the more surprising things of the research in general was how much of an impact herbivory was having relative to lily leaf spot, which is kind of what we expected going in.
So in relation to that, do you think that it might make sense to actually put deer enclosures around some parts of these populations to keep them viable, seed producing?
Yes, definitely. Actually, one of the things that we spoke with the Southern Appalachian Highlands Conservancy was maybe trying to put like tomato cages around those roadside plants to prevent mowing. And then as for deer exclosures, we actually had some great help from Dr. John Davenport's wildlife biology class last year. We have 2 5-meter by 5-meter, so 25-meter squared deer exclosures up on Tater Hill, one in a forested population and one on a grassy bald. Unfortunately, Tater Hill did not do very well in reproduction this year. There was only a single capsule produced, but that one capsule was in our deer exclosure on the bald. So kind of very preliminary research on that so far, but it's hinting towards, yeah, deer exclosures are a good idea.
Do other animals like rabbits also eat the lily? Because last time I'd been up there, I've noticed a lot of rabbits.
I don't know about rabbits specifically. I know I saw a lot of insect herbivory, specifically on capsules on my second visits. I would suspect rabbits eat it, but that's getting into speculation. But yeah, I assume they eat everything green, right?
All right. Are there any more questions for our presenters? Not hearing any. So I would like to thank all 4 of our students for doing an awesome job presenting your projects here. I think that gives us a great overview of what kind of projects the Native Plant Society is funding. So for everybody else working on plants, looking for money,